Welcome to the BEL webinar series for 2023. We are excited to share new research and ideas in the neuroscientific community. Today's webinar, Sleep and Neurodevelopmental Disorder Biomarker Discovery at NIMH IRP, is from our presenter, Dr. Ashira Buckley, MD, is the Director of the Sleep Disorders and Neurodevelopmental Service at Intramural NIH. Please write your questions into the chat feature, and we will answer them during the Q&A period. Thank you for attending our webinar. Please visit our website, bel.company, for upcoming webinar invitations, or visit us on social media channels.
So this work that we're going to talk about this morning and hopefully have a nice discussion about is questions that I've been asking for the last couple of decades and what we've been able to do at the IRP with our group. So I always like to start with the NIMH mission. Of course, I work in the Mental Health Institute. Sleep, of course, is, transcends all different disciplines, but that's actually where we began and that's where we still are. I'm a neurologist by training, but I absolutely believe in the vision, which is to envision a world where mental illnesses are prevented and cured. And we're working to transform the understanding, and that's where sleep comes in, and treatment of mental illness through basic and clinical research. This is my disclosure slide. I'm going to talk about some research that funding that we got. None of that came to IRP, but we do currently have an understanding, a memorandum with BEL. So those are my disclosures. I never start a research talk without talking about the children and families who participate in our research, without whom we have nothing to talk about. And my fantastic staff on the left, Sleep EEG and Sleep Labs are 24/7. And so these guys really work incredibly well together to make that work seamlessly. So sleep and neurodevelopment biomarker research, what does that mean? How does it work? And I think to frame it, really is just to say that we're always looking for evidence of subclinical or premorbid manifestation of sort of the worst behavioral disorder. So if we can get in early, that's always the thinking, right? Then we can prevent the downstream sequelae of living dysfunctionally. And so that's what we're looking for. Much of my research started with ASD, just kind of through dint of being a child neurologist and looking at sort of a lot of questions that were unanswered. And we started collecting sleep metrics, and we'll talk about what that means. That's a very big, broad category. But this diagram I really like, because anyone who knows anyone who does clinical research in neurodevelopmental research, and by that I include schizophrenia, the severe mental illnesses, so basically neuropsychiatric research, you know that sleep disruption is really integral to that. It's prevalent, it's ubiquitous. It's somewhere between 70 and 85 or maybe 90% of folks who have a psychiatric disorder will also have sleep disruption. And so I love this figure from MICROARCHITECTURE because it really lays out how the approach has been framed, right? So this says ASD in that little bubble on the left, but I could say schizophrenia, it could be depression. And the idea is, does the neurodevelopmental or neuropsychiatric disorder create this dysregulation that then impacts sleep, and then it's this terrible chicken and an egg syndrome kind of thing? Or are the sleep disruptions and the behavioral manifestations sort of two different sides of the coin of the same intrinsic abnormality? And I think the field, and certainly our group, is moving much toward this understanding of that these sleep disruptions are intrinsic to the disorder that we're looking at, and that also lends itself to this huge investigative window about if we can sort of figure out where those disruptions are coming from, can we walk them back and tie them to the disorder? And so there have been a lot of folks who've been looking at this question in its various different forms. And in terms of ASD, for those of you who are very interested in the history of ASD and sleep research, this is a very generous article written by Chloe Williams at Spectrum News out of the Simons organization. And she covered our lab and some others about a year ago now. Everyone who's sort of working in this space and the different areas and expertise that they bring to it. For instance, you can look at through a genetic lens, and Jonathan Lipton at Harvard, and looking at clock genes and tuberous sclerosis and ASD, to our work looking at EEG. So there's a lot of different places to enter this conversation. This is a slide that I love. It's from one of our sleep mentors, who's also an incredible epileptologist, Greg Holmes. Basically, what he's showing here is, you can enter this conversation at any particular level. I like to just always point out that we are entering it here at the microcircuit level, trying to get as proximal, as close as we can to the behavioral phenotype. And of course, then working with our colleagues who are having the conversation at a different level is the most important thing. And again, that little picture down at the bottom there with autism, you could substitute any neurodevelopmental disorder for that. Okay, so how do we measure and analyze sleep disturbance? For most of you, this is a recap. Most of the data that's out there is subjective. We use questionnaires. You're going to ask the parent or the caretaker, or the person themselves. Objective measurements are where we've been focused for a couple of decades. I'd like to always mention that actigraphy is a proxy for sleep-wake. It gives valuable information, but it isn't a substitute for the gold standard, which is polysomnography. So we're really interested in oscillatory information, and in particular, neural oscillatory information. It's important to marry that with the other physiology, and there's a little picture of our friend there having a PSG, but we're focused on the neural oscillations. Because when we record from the sleeping brain, these states, as you know, then can be further divided by differences in frequency, amplitude, morphology, and those are reflective of underlying neurotransmission. And so we never forget that, because they not only represent differences in sleep depth, which is sort of, the first thing that people think about, light, deeper sleep, but they probably also, more importantly for us in neurodevelopment, represent differences in sleep function. So I'm going to spend a little bit more time on this slide if it lets me. So what we realized sort of early on was that the more difficult it was to recognize these morphologic changes in stage sleep, the more likely we were looking at a child who had a neurodevelopmental disorder. And that's a really very interesting concept. And I know this has been done by other folks looking at schizophrenia in adults. But in our world, this became very, very salient and sort of the platform that we began our investigations for. So I just want to note that, of course, these major oscillation classes are preserved across species. And the preservation of these bands suggests that their functional role is critical for survival. And therefore, deviations from these rhythms could potentially hold a lot of information. And that's really the thrust of our research. What information are we leaving on the table when we don't evaluate them? Each of these bands provides specific time windows, which neurons and brain structures can then interact. And so these, sorry, I lost my place here a little bit. But what I want to say is that because each neuronal structure can oscillate at different frequencies, that adds computational power to the brain. And so neurons can oscillate at different frequencies, each being synchronized with different neuronal oscillations in a different structure, right? And these mechanisms allow individual structures then to set up these communication channels. And so when we talk about sleep EEG, we are holding all of those concepts, and we're using the EEG as a way to neuroimage the brain or reconstruct these communication channels, if you will. These are some of the tenets of developmental sleep research that we hold, that many of these metrics, and I'll show you a little bit of that in a bit, follow an expected maturational course, that they both reflect and contribute to cognitive and behavioral function. And putting those two things together, they offer they offer to us an accessible biomarker for diagnosis and treatment. And again, we use the sleep EEG because it's robust, it's reproducible, and ultimately it's a scalable tool for development. It makes it very exciting. So the two protocols that we're going to discuss, some of the research that we're going to present today, are from Subtypes, which was done about 15 years ago now, and Toddler, which was closed about 5 years. And you can see the little diagrams here, but just the take home from these are that this Subtypes protocol, the average age of these kids was about 4.5. And we had the ASD group, a little over 100 kids with autism. We had their typically developing controls, but then we also had a group of kids who were matched for nonverbal developmental quotient that didn't have autism. So we had this other interesting cohort to compare and sort of be able to ask questions about intellectual disability and how it's related to ASD. And keep in mind, our group is very different from a lot of ASD research in that all of our children with ASD also had intellectual disability. So that's the subtypes. We're going to talk about that. And then Toddler. Toddler is a little different. Each child had not just one, but 2 EEGs, about 18 months apart, and they were a lot younger. So these children, their average age was about 1.5 years. To come into the clinical group, you just had to have a flag where your language was delayed. So these are children pre-diagnosis, about 1.5, that we followed for a couple of years with a baseline and a follow-up compared to their typical counterparts. So those are the two studies we're going to briefly talk about. I just want to, you guys all know this, that there are many, many different ways that you can start to evaluate and investigate the sleep electrophysiologic metrics. You can look at architecture, power, coherence. We're going to talk about some of these things in terms of macro architecture, total sleep time, latencies, percentages, and then the micro architecture, coherence, and the microcircuits. So really early on, we published a paper saying, look, we were finding these gross macro architectural differences in autism. The panel on the left just sort of highlights one of the things that we were most interested in, which is this relative REM deficiency that we found in our analysis in these children who had ASD. They also had increased slow wave sleep. There are questions about homeostatic control in there that we haven't been able to explore yet. And of course, the most reproducible change, which is that children with autism tend to sleep about an hour less than their counterparts, and that's been reproduced over and over again. Okay, but then we want to look deeper at some of the microarchitectural details. Many of you are probably familiar with spindles. They have this unique neuronal rhythm. They have a particular morphology, a particular oscillatory frequency. They're either from 10 to 16 or 9 to 15 hertz, depending on who you read. Many people break them down into slow spindles and fast spindles, with fast spindles being centrally located and the slow spindles being more in the frontal region. There are lots of different ways to evaluate spindles, and some of these metrics here are listed on the bottom. I'll let you read them. And then there's a newer one that we've been looking at, which is called CHIRP, and it's the intraspindle frequency change, and which is really thought of as the morphology of the spindle. And what does the deceleration of this look like? And so that's we're going to get back to that in a second. The reason that we're interested in this in neurodevelopment is that it represents this little volley, this little crosstalk between the thalamus and the neocortex, mediated here by completely GABAergic innervation from the TRN. And so the health of this microcircuit was most frequently studied prior to our getting involved and other folks sort of also looking, was most heavily studied in schizophrenia. And it had been documented by Fabio Fararelli, who's now at Pitt, and Dara Minogue's lab, that there was a spindle deficiency, even in medication-naive folks, people who had schizophrenia. So for a long time, maybe people thought that this was a unique marker for schizophrenia. It also obviously is related to sleep depth and protection of the sleep process, but also this interesting relationship with a neurodevelopmental disorder. So we wanted to look and see, well, what did that look like in our population? So we went back to subtypes, that group of kids who were 4.5. Remember, there were 3 groups with autism without, but matched for nonverbal intellectual disability, and then the typical kids. And here we found that there was a spindle deficit in the children who had autism. So then Dr. Farmer, who did this work with us, did an exploratory analysis suggesting really that this spindle count couldn't entirely be explained by the intellectual disability, and that there was potentially this unshared portion here. This was the Vineland Socialization Score, by the way, for people who are interested, that potentially there was a relationship between behavior and not just intellectual disability and spindles. So again, it was a small N. The study really wasn't designed for that. This was an exploratory look, but it helped focus our efforts to try to understand what more might sleep tell us, and what questions do we need to ask to continue to parse this out. Just very quickly, we looked at spindles also in the toddler cohort, right? So now we're coming younger and we're looking at the kids who are 1.5 years. This is just to remind you that the age and the relatively small ends here. And these are the leads that we looked at. I'm just going to show you a couple of panels. This first one is the density of spindles baseline versus follow-up for all children. And then so the baseline, slow spindles, the faster spindles, baseline of all kids, the follow-up, and the difference. So the take homes from this is just that the density of slow spindles decreases from baseline visit to the follow-up and the central leads. I think you can see them here. Density of slow spindles increased from baseline visit to follow-up in the frontal channels. And the density of fast spindles decreased from baseline visit to follow-up across multiple channels. So all this is doing is just sort of plotting the change. What does the change look like in our group? What does the trajectory look like? And then you can break it up into developmental cohorts. So for instance, we can pull out slow spindles. We can ask the same question kids who have language delay related to kids who have typical development. What is the change here? Increased more from baseline to follow-up in the TD group than in the LD group. So is there a maturation here that's happening that's not happening in the LD group for slow spindles in the frontal cortex? And if that's true, what does that mean? And then again, you can just go and you can pull out and you can say, okay, well, I want to ask the same question about fast spindles. And here's what we found here, basically just showing that there was a decrease in these spindle to a lesser extent from baseline to follow up in the LD group. So is there some type of catch up happening? Again, these are just sort of characterizing what these developmental groups are doing vis-a-vis a particular metric. And it just brings us to more questions. Okay, another example of a microarchitecture that we've been interested in is coherence. I think you guys probably all know just a general understanding of coherence, this estimate of the degree of connectivity, spatially separate clusters, and highly coherent nodes are interpreted as having substantial temporal coordination and somehow they're related. Low coherence then interpreted to mean that this activity is pretty much autonomous. There's been a ton of work in ASD and coherence. Most of it's been done in the awake state, most of it's been done in functional MRI, and the results differ by approach, by cohort looked at. We wanted to look at it with our group, and that's what I'm showing you here. And the take home from this is this heat map here of the kids in subtypes. And this heat map is comparing typically developing children to children with ASD with about 171 different electrode pairs color-coded for region with the canonical frequency groups across the bottom, awake, REM, and slow wave sleep. And the take home here is that when we looked in the awake state, we didn't find any changes. But when the child went to sleep, when the children went to sleep, we saw evidence for increased coherence only in the ASD group and in these particular regions, frontal parietal pairs during sleep. So is sleep the most sensitive window to look? We're not sure what those increased coherences necessarily meant. We wanted to look at that in our next group, in our going younger, to the kids who don't have a diagnosis but have a language delay. We use those same pairs here, and you can see them out. And my mouse to work here, based on the frontal parietal changes that we had seen in the previous work. And they're plotted here, awake, REM, slow wave sleep. In the dark blue, you can see that this indicates higher coherence for these language delayed children than their typical counterparts. And in these particular patterns, here are the lead pairs, here are the frequencies. So again, able to separate, look at differences, but what does any of that actually tell us? So we wanted to ask exploratory questions. Here's a separate exploratory analysis. We evaluated the relationship between coherence and behavioral outcomes using verbal developmental quotient and nonverbal developmental quotient, and then the Vineland socialization standardized score. So in this panel, and this is work done by Natalia Kuzmienko, who's a fellow at Harvard at NSRR, and we'll talk about that repository in a little bit, and our senior research assistant, Shanine Hong. And in this, you can see this is REM sleep, slow wave sleep, language delay, and typical development. These are the lead pairs that we talked about. And it turns out that the lower the verbal score, and if you have language delay, the higher the coherence in REM sleep. We didn't find anything in slow wave sleep. And if you have the lower your verbal score, if you have typical development, higher your coherence in this completely different pattern. So these are things that we're very interested in pursuing in larger numbers. Again, these are short exploratory pilot studies. Most of these studies are less than 100 kids per cohort, as you've seen. But the real exciting part of this is to understand that there are so many questions. And our take home from the last 15 years of this research has been that sleep is woefully underleveraged for neurodevelopmental biomarker discovery. Many more questions than we have answers, a lot of avenues to study. And so I want to just bring up this very recent endeavor with Sean Purcell and Susan Redline, who are the co-PIs of this national sleep research repository, which I'll talk about, and Natalia Kuzmienko, who I just introduced you to as the fellow up there, where we wanted to look at a large cohort then of sleep EDFs or sleep tracings and see if we could start to map some of these neurodevelopmental changes. So I'm going to give you the punchline first, which is that overall our results indicate that this sleep architecture, as we thought, offers a pretty sensitive window for characterizing brain maturation, which of course then suggests potential for more scalable, objective sleep-based biomarkers. The cohort that we're talking about here is Nationwide Children's Hospital. A couple of years ago, they deposited about 3,500 PSGs, 34 unique subjects. They're broken down here for you to see. Between 0 and 20 is the age group. You can see the number of males and females, and these are the leads that they're deriving the information from: F3, F4, C3, C4. So with large numbers like this and across the age span, you can start to look at developmental trajectories, right? So what does N1 duration look like as you mature? Again, these are cross-sectional, and they're that's a clinical population, so it's not perfect, but it actually gives you here's a very nice validation from this data. We know this from other research from Dr. Feinberg and his work in adolescence, that as you get older, your slow wave sleep in minutes decreases. So these are just nice checks to show that we're showing what's been shown before in terms of developmental trajectory for these large macro architecture questions. You can do the same thing with percent time in each state, right? These are some nice curves here. And then you can start to get more refined and you can say, well, I want to know what does the slow oscillation rate look like? What does fast spindle density look like? These are just examples of starting to probe these large data sets for information about microarchitecture. You know, at some point you can envision, you want to know at age 4, 8, and 15, what does the trajectory of spindling and slow oscillation coupling look in ASD? So we're hoping to set up these databases to be able to be queried that way for hypothesis generation. So now I want to take you to some of the exciting work in this paper, the mapping neurodevelopment work. And by the way, this is available in bioarchives right now. It's about a 40-page paper, and you can go in and dig to all the different things that we looked at. And I'm not going to talk about all of them. But the idea here was you should be able to map out some of these micro and macro architecture metrics in terms of development. For instance, we found that the strongest macro metric was the number of non-REM cycles. That was the strongest predictor, not total sleep time or latencies, but the number of non-REM cycles in terms of age. And the strongest micro was actually that spindle morphology, or the chirp, the rate of deceleration in that spindle. That was the most tightly tied to age. So those are interesting things that you wouldn't necessarily predict. But you can also start to ask more comprehensive questions related to tool development, really. And what Natalia did here is she wanted to know, could you use this as sort of a brain aging tool? So brain age has been assessed in other modalities, mostly in adults, but it's been done in Alzheimer's and schizophrenia, mostly with MRI. Sleep EEG, we believe, can give comparable results, and there are a couple studies in adults. So we wanted to look at this in pediatrics for this developmental trajectory in brain age. So based on the metrics, some of these major metrics, and again, there's 40 pages and show you all the micro and macro architectures that she used, those metrics, but she constructed a model to predict an individual's chronologic age. And the approach was initially to remove people with neurodevelopmental diagnoses and then validate, and this is the validation sample, you can see how many she used, against separate studies of children who came into the repository for other reasons. So these two groups of kids were ruled out for any neurodevelopmental disorder. They came in because they had obstructive sleep apnea. And so she validated them first against the training set and then a testing set within the Nationwide Children's database. And you can see the R here was 95. And then against the CHAT study, which was a study of about 1,000 kids in an SRR, this was 0.65. And this is probably because this age group was a little more limited. And then in a more comparable age group in the PAT study, another study of OSA with kids without NDD, and a pretty good R here. So when she did that, then she wanted to actually look and see, well, how does this stand up against neurodevelopmental cohorts? And that's this right side of the slide here. And the take home from this is that like would be expected, there was a greater variability in the neurodevelopmental cohorts. And you can see we looked at ASD, ADHD, intellectual disability, Down syndrome, CP, and epilepsy. And granted, there is some overlap here. And these are all from diagnostic codes that were sent from Nationwide Children's. So again, this is retrospective clinical data. But what was very interesting here is that kids with intellectual disability, or kids with Down syndrome in particular, this was the furthest away from being able to predict their age from this model. And I believe it was about 2.2 years discrepant here, younger than would be anticipated, and about 1.7 years here, younger than would be anticipated. So we're just going to pull out some examples to illustrate some other important concepts about this. And again, you can look at the full paper. But the developmental trajectories of different sleep parameters, they're distinct across the lifespan. So, for example, to compare, and I'm going to orient you guys here in a little bit, to compare further these metrics, then we can pull out the adult cohort samples in NSRR. And there are lots of thousands, tens of thousands of data points that Sean Forsell has collected, mostly in adults, and they go up into the 80s. So on these two panels, you're going to see from 0 to 80, 0 to 80. This one is spindle density, changes across the lifespan, and you can see it's not a linear trajectory. If you take this and you look at the fast spindle density at C3, for instance, and you go across the lifespan, and this green line is the mean from the Down syndrome cohort at NCH. And you can see that their spindling density looks much more like a 75, 80-year-old, the mean here, than anywhere else, even though it's a pediatric cohort. But if you look at another metric, in this case, absolute theta power changes across the lifespan, then the Down syndrome group maps up here, which is more like age 6. So you always have to be careful about what you're looking at. So when we're looking at brain age, or like a composite metric like Natalia is experimenting with, overall, it might look like the brain age is younger, but not maybe for every metric, and that's giving us clues as well. The other two panels I want to briefly touch on here, this is just a nice little check for our group, and this is slow spindle density at C3, and then the degree of intellectual disability across the bottom. And basically what this shows, and has been shown before in a couple of other studies, is that the more profound your intellectual disability, the lower your spindle density. So that was a nice check. And then this one here, I think, has a lot of potential hypothesis generating interesting questions here. This is changes with age and Down syndrome versus the rest of the sample. This is the coupling overlap of slow spindles with slow oscillations at C3. And so what's interesting here is that the relationship of all of these oscillatory parameters was deeply important for understanding learning and memory. So not just a single metric, but how does that oscillation relate to other oscillations? And of course, these are all questions of timing. So the slow spindles with the slow oscillations, hippocampal ripples, all of those things need to be lined up in a particular way to optimize learning and memory. So you can start to ask, what does it look like, the rate of coupling slow spindles and slow oscillations in the whole group? And you can see that as you get older, that overlap increases in the rest of the non-DD sample. If you plot Down syndrome against it, they actually have a declining slope here. And so as you get older, the gap here is exacerbated. Other folks are sort of optimizing, and here it looks like you're losing ground. And so this gets bigger and bigger as you get older in this age group. And of course, there's lots of questions that you can start asking about that. So these are just some of the examples of how we probe this data. And then for the last 10 minutes, I'm just going to posit some ideas about how we move forward with some of these questions closer to mechanistic understanding and closer to therapeutics. And there are three general tenets, three general things that we've been following: consensus building, using our sleep data repositories and contributing to them, and investment in infrastructure. So we started this work almost a decade ago now with the intramural with workshops, where the 2014 one was about a dozen people from different fields asking, what don't we know about sleep and neurodevelopment? And the answer was a lot. So in 2017, we had our first formal convening, and that was more like 50 people. And that link to the proceedings is available at the NIMH website. And really what we said is we really need to build an infrastructure. We need to find a way to get those extant data sets out of the shelves of pediatric centers of excellence and into data repositories. That was followed a year later by a workshop that was just about how do we develop the best animal models to answer this question. And then about 18 months ago, symposium devoted almost entirely to what don't we know about the first year of life and the way that sleep builds the brain that we need to know. And so over this period of time, you can see just if you're going to measure by rate of publications in this area, there has been definitely an increasing interest. We're very happy to see that. I just want to put this up here for those of you who are not familiar with the National Sleep Research Resource. This is what I was just talking about with the NCH database. They deposit all of those and they allow us to sort of go in and take a look. Those are freely available data. This platform was built for these types of queries, and it is co-PI'd by Susan Redline and Sean Purcell. In 2019, we got a research grant from the Pediatric Epilepsy Research Foundation to get those extant data sets off of the shelves of these pediatric centers and into NSRR. And that has allowed a further understanding about diagnostic differences. A lot of these centers have different areas of expertise that they're collecting data from. And then this is the sort of culmination of where you want to go with all of these retrospective collections, which is a longitudinal prospective study. This is an accelerated longitudinal prospective protocol that I'm the PI of at Intramural, where we marry all of this data, behavioral, genetic, psychosocial, to the contemporaneous EEG signals. And we see these kids over time to build this developmental trajectory. What that allows us to do is follow children like these. These little Spideys are each 6. They're from very similar neighborhoods in DC. They each have a parent with schizophrenia. This child has social, affective learning deficits, and this child does not show any signs of that at all. So these types of collections and longitudinal assessments allow us to get closer to mechanism, or at least to developmental trajectory and what might be different between these guys. This is just a recap of what we've been doing for the last 15 years, which is developing the standard to get this information, collecting the data, archiving it, and ultimately the idea is to just have data sharing. Nationwide Children's we talked about. Right now, between all of the studies, these centers, we've got about between 17 and 1,800 now studies there. So 6-7 years ago, there were less than 100 kids in NSRR, and now we have over 6,000 and growing. So we're happy about that. We're about 3/5 of the way through with our pilot. This is enrolling the longitudinal prospective data collection. And we'd really like to expand. That's really where we're going. Just this year, Northwestern joined our consortium from Phyllis Z and Circadian. She has the first circadian medical clinic established there, and she's a wonderful giant in the sleep field, particularly in circadian rhythms. So we'd like to be able to ask these questions, social determinants and oscillatory sleep metrics. We didn't touch on that at all. This is a very important area that we're interested in. We didn't really talk a lot about epilepsies and the canonical rhythmopathy, right? Pediatric epilepsies. How do we begin to evaluate these questions? Well, we do that by continuing to build our consortium and thinking about how to scale these assessments. And one of the very most important things that we want to do is really always keep in mind that the point of care devices validated in lab or however need to be developed alongside the hypotheses. And that's why we're very excited to partner with BEL, because we have a lot of questions that we need to be working with partners to be able to answer accurately. What about increased REM coherence in kids who have LD? How do we start to unpack that and understand why their sleep process is different from their counterparts who don't have language delay? Because these rhythms carry really important information about cognition and behavior. We are convinced of that. And so partnering with BEL will allow us to ask these questions as they did about where are these oscillations coming from. So better identification of the source of these will be helpful. And ultimately, the idea, of course, is to lead to therapeutic opportunities in keeping with the NIMH mission statement. So I just have some ending thoughts, and I appreciate your staying with me for 40 minutes. And that is that one, sleep neurophysiology is underleveraged for neurodevelopmental discovery and therapeutics. Two, you have to absolutely do this with multidisciplinary teams. You have to have engineers. You have to have computational folks. You have to have psychiatrists and neurologists. And that this part is what we've been working on, and this needs to continue. So with that, I'm going to stop talking, and I'm happy to answer any questions or just engage in conversation with you all.
Thank you so much, Dr. Buckley. That was really interesting talk. We do have a couple of questions here in the chat. And I just want to encourage everyone to go ahead and post their questions into the chat if you do have questions. One question I just wanted to clarify the repository for the papers that you were mentioning. And that was great. Yeah. Is it the sleepdata.org? Is that the correct?
So this paper is in bioarchives right now. You can go look at it. NSRR is the repository. And I think people should just go to sleepdata.org and play around on that.
Great. And I think I did see a question pop up about what were the different sort of methods that you used. I mean, you mentioned a lot of different sleep metrics, but were there a variety of methodologies for doing the sleep data?
For doing the sleep data, you mean for the plots that I showed from Natalia?
Mm-hmm.
Yeah, so I'm going to refer you to the paper, and it's 40 pages, and she does actually go through all of the analyses and the approaches. It is a ton of data, right? So it's so much information. So we do this in conjunction with the statistical core at NSR. But yes, I'm going to refer you to, rather than misspeak or tell you guys something that isn't true about how we derived any of these, especially her composite metric. I'm going to refer you to the methodology in that paper, and it's available for you guys to go through.
Great. There is a question that asks, can we talk a minute about data harmonization?
Yeah.
Integrating across retrospective analyses?
Yeah. And so the reason that I love that question is because, we had this idea to get these extant data sets for a long time, but until 2017, when we met Sean Purcell and we learned about NSRR, which by the way is funded by NHLBI, because of the harmonization piece, we didn't in our little shop, right, have the ability to do that. That is something that they do at NSRR. And so you're right, it was it depended on the site that the data came from. So for instance, Boston Children's Hospital, they had a very similar way of acquiring and cleaning data, and that was an easy lift. Geisinger Medical Center was a different beast altogether. And so that those had to be cleaned at NSRR and harmonized to be able to even begin to speak intelligently about. And I didn't show you any of the perf data, and that's the retrospective data from the 5 sites. I didn't show you any of that. But yes, a fair degree of cleaning and harmonizing had to happen. And NSRR, that's what they do. So that took many months of coordinating different folks. And it was a heavy lift, but now that the infrastructure is in place, we're ready to go much, much faster.
That's great. I just have a couple questions. well, I'm going to skip to this other question. How might interventional studies best investigate potential changes in sleep? Do you recommend analyzing at the micro and macro level when possible?
Yeah, and I think there's room for all approaches. That paper that I just showed you, using a single metric to say evaluate the brain age gap. There are obviously big detractions from that, right? That's why I showed you the second slide about how, yes, maybe if you take that single metric, you might end up somewhere on the graph that says that this person is this age. But when you start to sort of unpack the different microarchitecture metrics, for instance, those represent different microcircuits. And so I think you sort of have to be careful with the one metric approach, and that combines macro and micro. What's exciting about databases, repositories coming together at NSRR is that for a long time, the study of sleep research was just macro, right? Whether it was narcolepsy, and that's a wonderful success story, or depression and REM latencies, we really didn't have the computational power to take these enormous data sets and even begin to start asking how they all come together. So I think it really depends on what your interest is. It depends on if you can marry one of those metrics to a real functional outcome or even a mechanistic hypothesis. So I think there's room for all of those things, and I certainly don't have the answer. Just more minds on the wealth of data that's in the sleep EEG and sleep neurophysiology is really our goal here.
That's terrific. I will just go ahead and thank you again. And there is a follow-up question. Isn't it much harder to do this in a large, uncontrolled collection of data sets than in a smaller, well-focused set?
Yes, I guess. It depends on what your question is, right? So again, we're IRP, so we're a small group. We have in-house lab for everybody, and we can ask very specific questions that are very focused. But ultimately, in order to make this relevant, you need to be able to think about how to translate that information from sort of small, well-described groups into scalable endeavors, right? I mean, I think that's really what we're trying to do here. Not everyone's going to be able to come in and get an in-lab validation. So like all science, right, the better controlled you are, sort of the purer your result. But psychiatric disorders and neurodevelopmental disorders are messy. And so when you start to constrain and you're sort of well describing something in a small group, I'm not sure how well you can extrapolate that. So we do need the vision to be able to figure out how to expand that in a way that makes scientific sense.
Great. And we have another question from Dr. Farrelly. Do you think there is an opportunity to run NIMH-funded multi-site studies to characterize sleep, neurophysiology, and neurodevelopment?
Is that Dr. Fabio Farrelly?
I believe so.
Yes.
Hi, how are you, Fabio?
Good, great talk.
I was giving you a shout out. Obviously, yes. I mean, I think that part of why I, travel around the country and the world giving these talks, and it has paid off on certain levels, is just to put the spotlight on how much more we could do if we concerted our efforts. If we continued to build on this infrastructure and said, look, this is a very rich avenue for mental health discovery. And so that's basically what I think about every day, is how to scale that up. And I would hope that NIMH would reopen their interest. I think we've gotten some inklings that they are. But yes, that is the goal.
Okay. And I think there is one more question about looking at data from infant siblings of children with autism to try and find biomarkers.
Yes. What's that question? What is the question?
It's just a kind of general comment. It's saying that would be a really interesting way to look at data.
Yeah, and for a long time in the autism world, the baby sibs was very hot, right? For anything, not just sleep EEG, but the siblings, because they had an increased risk. Yes, I agree. That would be interesting. What we're studying with ESP, we wanted to specifically broaden that out. And again, kind of referring back to the previous comment, you can answer some things very well with small, well-described groups. But we want actually to be looking at the full trajectory of neurodevelopmental disorder. And so, for instance, the study that we're looking at with toddler, with kids who have language delay. Language delay is a huge flag to go on and develop autism, but not every kid with language delay is going to do that. 9 of those kids did, by the way, end up doing that. So baby sibs is a good way to enrich your likelihood that you'll end up with more kids with ASD. And there are folks who are very devoted to that. Our approach is to be much broader, because we're looking at functional outcome regardless of diagnosis. And so it doesn't have to be ASD. It can be any functional outcome, broader, so language or social deficits across diagnoses. So that hasn't been our approach, but it has been one that has been adopted by many folks.
All right. Well, thank you so much, Dr. Buckley. I think we can go ahead and wrap up your talk here. And I want to thank everyone for joining. And just to let you know, we will have this talk. It has been recorded, and we will have it on the website for people that couldn't join or who joined a little bit late. We will have it so that you can see it at a later time. Thanks so much to everyone for joining, for Dr. Buckley. We really appreciate it.
Bye-bye now.